everyone, and welcome to the first of two lectures of the GL4U RNAseq module set. If you are listening to this lecture, I assume that you have already completed the complete GL4U introduction module set consisting of 4 lectures and 4 hands-on activities through Jupyter Notebook. So if you have not completed the GL4U introduction module set in full, please go ahead and complete that before continuing on for this GL4U RNAseq omics module set. So this lecture is going to cover RNA sequencing and data processing of RNA sequencing data. The topics covered here, I've split into 3 parts. After each part, I'm going to have you pause this lecture and do the corresponding steps in the hands-on activity Jupyter Notebook that covers the same steps that I go over here in the lecture. So for part 1, we're going to go over what came before RNA sequencing, followed by an overview of an RNA sequencing experiment. And then we're going to get into the RNA sequencing data processing pipeline, starting with raw data QC, followed by trimming and filtering those raw data and performing a subsequent QC or quality control on the filter data. We'll then pause to give you time to take the first hands-on activity of the GL4U RNAseq modules. We'll then carry on with the lecture, the part 2 of the lecture, which is going to cover RNA-seq data processing, picking up at the alignment step, followed by QC of those alignment data, and then assessing strandedness of our aligned reads. And then part 2 will finish with us talking about how we quantitate our RNA-seq data and then perform quality control on our quantitated data. You'll then have time to jump back into the RNA-seq processing Jupyter Notebook to finish those steps in that notebook of the hands-on portion of the GL4U RNAseq module set. You'll then come back here for part 3 of this lecture, which will cover beginning with analysis of the RNA-seq data, starting with normalization of those data, followed by differential gene expression analysis. And lastly, we'll briefly touch on the data visualizations that you'll be generating in the hands-on portion. You'll then hop to the GL4U analysis portion of the hands-on part of the GL4U RNAseq module set. So starting here with part 1, what came before RNA sequencing? Polymerase chain reaction, or PCR, is one of the first ways that we were able to duplicate select pieces of DNA. This was an invention or discovery by Kary Mullis, depicted here, who went on to win the Nobel Prize for this work. And in PCR, you start with your DNA template of interest, as you see here. So the region of interest here is going to be depicted in darker blue, and then you'll need to find a region flanking the region that you want to duplicate, that we're going to refer to as our priming region in light blue here. You'll then need to add to this reaction primers that are going to be complementary to this priming region on your template, as well as dinucleotide triphosphates, so your A, T, Cs, and Gs, and your enzyme polymerase to be able to build a strand from a template strand. PCR takes place in 3 steps of each cycle. In the first step, the DNA, the double-stranded DNA template, is denatured. So this is done just by heating this up to a high temperature to break apart those hydrogen bonds, holding the 2 strands together. This then exposes that priming region that's going to be flanking the target that you want to amplify. And then we're going to lower that temperature and make these conditions efficient for this primer to be able to hybridize to its complementary region on the target. And this is going to happen both on your sense as well as your antisense DNA strands. Once those priming regions are hybridized, you're then going to go into step 3 of the PCR cycle, which is the extension step, So the hybridization of that primary region will recruit your DNA polymerase here, which is then going to pull in those dinucleotide triphosphates to build the complement strand from that target. Again, this is happening with both the sense and the anti-sense strands of your template, so that at the end of this first PCR cycle, if you started here with one piece of your DNA template, you will end up with 2 copies of that DNA target region. This then repeats for multiple cycles, depending on how many cycles you set up your PCR instrument to perform, so that by the end of the second cycle, you will end up with 4 copies of your target region. By the end of your third cycle, you'll have 8 copies, and so on, until you get 2 to the nth copies, where n is the number of cycles that you perform. And if you guys want to watch this in a video, I linked out here to a YouTube video that shows how PCR reactions work. I recommend you check that out. So next that came is we wanted to find a way now to be able to quantitate how much of, let's say, a gene was expressed in a sample in our treatment group compared to a control group. So we needed to find a way to quantitate the number of transcripts of a particular gene. And so in this example here, we're going to start with messenger RNA, and this process is known as reverse transcription quantitative PCR. And so starting with this messenger RNA here that we isolated from our samples, the first thing that we are going to have to do is convert that RNA into complementary DNA, or cDNA. We're going to be using an enzyme called reverse transcriptase, which I believe was hijacked from viruses, to be able to perform that conversion. And then we'll have our cDNA, and this is going to be used as your DNA template into a PCR reaction. So it's going to have the same reagents that we saw in the previous slide. However, we will additionally add in this type of RT-qPCR here, we're going to add in a molecule called cybergreen. And cybergreen binds to double-stranded DNA. And then once you excite it with light, it will release a fluorescent signal that we can detect. And so every time you're starting cDNA, your region of interest is copied. In that last step of the PCR cycle, the cybergreen will bind to that double strand cDNA of your target region, producing a light signal. And so the idea here is that there's going to be some background fluorescence that's going to be detected, but as you produce more and more copies through more and more PCR cycles, that signal from the cybergreen that's bound to the double-stranded cDNA in this case, that signal is going to get brighter the more cycles you have. And the more of your gene that's expressed in your sample to start with, so the more that you have to start with, the sooner that signal is going to get brighter than the background fluorescence. And that's a way to quantitate how much of your target gene is there by seeing how quickly you're able to detect a signal beyond background noise. Again, the sooner that signal appears, the more there was in your sample to begin with. And you can do this alongside a standard curve of a target of known concentrations to get a rough quantitation of how much of your target gene was there to start with. Another way to do RT-qPCR is through something called a process using TaqMan probes. And so with this process, what we do here is, again, we're going to go through PCR cycles. So we're going to have a primer flanking our target region, or our target gene in this case. In addition to our primers, we're also going to have a third region that's going to be specific to the region of our gene that we are wanting to detect. And this probe here is also going to have a fluorescent signal on its 5' N and a quencher on its 3' N. such that when this probe is intact, the quencher is going to be close enough to this fluorescent signal to be able to suppress the signal through fluorescence resonance energy transfer, or FRET. And so what's going to happen here is once you denature your target template cDNA to have your two strands, your primer is going to be latching onto the priming region, your TACMAN probe to the center of your target. And then when polymerase is recruited on in your primer, it's going to go ahead and synthesize that complementary strand from your target, eventually reaching where your probe is and bulldozing through that, releasing the fluorescence because it will then be far enough away from its quencher to release a signal, which you will then detect that fluorescent signal. So similar to the cyber green assay, the more of your gene that's expressed in your starting material, the sooner that fluorescent signal is going to be detectable above the background noise. And so again, this is a way to be able to quantitate how much of your gene is in your starting material and a way to compare the amount of your target gene in a treatment sample versus a control sample. Of course, here you also want to be looking at a control gene that you know does not change in the context of the treatment that you're looking at. And that way, this way, the amount of that gene you can use to normalize for your gene of interest. And also, it's a really good idea to perform this RT-qPCR in triplicate. So that's technical triplicates for each sample, to have more confidence in your quantitation measurement. And so what this looks like is it'll get plotted here where the number of cycles is going to be on your x-axis and the amount of fluorescence detected is on your y-axis here. And this is just showing the background noise will be around here. And each set of lines is a sample in triplicate. And so as you see, during the PCR, that signal that's being registered after more and more copies are made will, after a certain number of signals, depending on how much you're starting with, that signal will become greater than the background noise as it undergoes exponential amplification into this linear amplification phase and will eventually plateau out. It'll reach the saturation point for that fluorescent signal. And so what you're going to look at here is determine what cycle number was the signal detectable beyond background noise in each one of your samples. And again, the sooner this signal is detected or the fewer number of cycles it takes to detect the signal above background noise, that means that there was more of your starting material in your initial sample. So a sample, this sample here for this gene is going to have more of that gene in that sample compared with this sample over here that took many more cycles to be able to detect a signal beyond background. So this was great. It was great for being able to measure how much of your gene you have in your starting material compared with a control, treatment and control. However, there are some limitations, and I do want to point out this technique is still very much widely used in labs today, but some limitation is that because you have to design primers that are flanking your target region, you will need to know the sequence of the gene that you want to quantitate, and so only annotated genes or genes in which we know the sequence of can be evaluated. You also have the potential for non-specific binding. So cybergreen binds to double-stranded DNA. However, it does not bind specifically, just any double-stranded DNA it will bind to. And so you will get some non-specific signal coming out of that cybergreen assay. It does require primer optimization. So you want to make sure that your primers are only attaching to a region flanking your target region so that you're not getting non-specific primer binding, and you want to make sure that your primers bind efficiently. So that does take some optimization. For the TACMAN approach, instead of having two conserved regions, the two priming regions flanking your target region, you're also going to need to find a third region that's somewhere in between your two priming regions that's going to be unique to your gene of interest for that TACMAN probe. And also, as I mentioned, this is going to require some housekeeping genes. Those are those genes that will not change the expression under the treatment that you're testing. So that way you can use the expression, the amount of expression of that gene to normalize for your target genes of interest. And also, as I mentioned, absolute quantification of the target gene is possible through using a standard curve being generated with a sample of known concentration. But that will also increase the likelihood of issues if you do not properly generate your standard curve. Also here you have a limited number of samples and number of genes that'll be able to be evaluated simultaneously. These can often be done in 96 well plates or even 384 well plates, but that still is a limit to the amount that you can look at, amount of genes you can look at, and amount of sample. Also, as I mentioned, all samples really should be run in triplicates, further limiting the N number that you'll be able to get per your group. And then if you can think of any other limitations of RT-qPCR. So one of the next techniques that came available to try to assess differences in gene expression between two samples is microarray. So just a quick overview of microarray. There are quite a bit of microarray data sets hosted on the Open Science Data Repository. And just to show you a quick summary of how this technique works, you will start with your treatment. So your samples of interest undergoing a specific treatment. So in our case, our ground control samples and our flight samples. You will then isolate, in this example, message RNA from your control sample and your treated sample. You will then convert that message RNA into complementary DNA or cDNA. And then this time here, you're actually going to label your cDNA with a fluorophore, in this example here. And it's going to be a different color fluorophore for your different treatment groups. So here we have our ground control that we're labeling in green and our flight samples that we're labeling in red. You then have a chip that contains these spots on the chip, and each spot has thousands of these probes. Probes are just short oligonucleotides, so ATCs and Gs, and these ATCs and Gs are going to be complementary to a known gene. So each spot here, again, this is only showing one, but really there's thousands of these probes for a particular gene. So each one of these spots would represent a different gene. And then you introduce your labeled samples onto this chip and then allow the cDNA from your samples to hybridize to these probes so that if the gene that is being represented by this probe here, if that is expressed in our ground control samples, then those samples will hybridize and release a green signal. If the gene is expressed in our flight samples labeled in red here, that cDNA will hybridize to the probe representing that gene, and we will get a red signal. And if that particular gene is expressed in both our ground control and our flight samples, then if it's in roughly the same amount, we would see roughly the same amount of green and red signal, which would come off as a yellow signal. And so these signals are detected by a fluorescent scanner. And ultimately, what you're left with here is a chip with a bunch of spots on it that looks like this, such that any area that is green would be that mean that that gene is more highly expressed in your ground control samples than your flight samples. If you have a red spot here, that means that that gene is more highly expressed in our example in your flight samples and your ground control samples. And any yellow spots here would mean that you're getting equal hybridization of that probe representing that gene from both your flight and your ground control samples. And so this is just a snapshot of what a chip looks like. And so here you see a range of colors. And so this really bright red color instance for here would indicate that this gene is expressed in our flight samples, but probably not in our ground control samples. Similarly, this bright green spot here is expressed in our ground control samples, but not our flight. This yellow spot would indicate that there's roughly equal expression of that gene in both our flight and our ground control sample. And then we have ranges in between there. So something more orange like this would mean that although this gene is expressed in both samples, it's probably more highly expressed in our flight samples. And then similarly, this greenish yellow here would indicate that this gene is expressed in both samples, but more highly expressed in our ground control than our flight samples. So again, this was great because you could have dozens of thousands of spots on each one of these chips. So you can look at 20,000 plus genes on a single chip here, which was great, and be able to determine of two samples which one had a more high expression than the other. However, this technology also comes with its set of limitations. Again, because the probes that are on each spot of that chip need to be complementary to the gene of interest that you're looking at in that spot. Only annotated genes, again, genes that we know the sequence of, could be evaluated. It also requires hybridization, and so that comes with a risk of non-specific binding. That means because although something that's complementary to the probe will bind, it doesn't have to be exactly complementary. So if a handful of nucleotides are not complementary to the probe, it may still bind enough to be able to release a signal. So you could get some non-specific binding and also cross-hybridization of sequences with high identity. It's going to be really difficult to detect splice isoforms, especially if your probe is for an exonic region of a gene that exists in more than one isoform. Then if you see that expression, you won't know what isoform is actually being expressed in your sample. Also, your dynamic range is going to be limited by your scanner. It's going to be difficult to calibrate a scanner that can be sensitive to simultaneously really low expressing or really low signal or low expressing genes to really high signal or high expressing genes. And also, once you get up high enough, you're going to reach your saturation point. And so this often ends up meaning that your low expressed genes are going to be highly variable, difficult to determine differences in expression for those low expressing genes. Also, this is designed to measure relative abundance. So you're measuring how much one sample is expressing a gene compared to another, but absolute quantitation, although you can do it using known standards, it is difficult to do with microarray. Also, not all fluorescent dyes have the same efficiency, so you might have unequal labeling efficiency in your fluorescent dyes, which you might assume means that there is less of a particular gene in a particular sample, but it could be because that dye that was used for that sample is just less efficient than your other sample. Lastly here, each chip that you have with your probes, you will get variation from one chip to the next. So in your first chip, if let's say in this chip you have your flight animal 1 sample compared with your ground control animal 1 sample, your next chip you have your flight animal 2 sample compared with your flight ground control 2 sample. You might get some chip to chip variation in there, and there are standards and ways to normalize and for that type of stuff. But again, it's just another limitation of the microarray. And there are others too. This is just listing some of them here. So after microarray, another technology came about called RNA sequencing, which I'm going to abbreviate as RNA-seq, pretty much moving forward. And this is a way to sequence RNA to be able to determine how much genes are expressed. Well, that's one use for RNA sequencing. So a quick overview of what an RNA sequencing experiment looks like. So here we have our samples of interest. In our case, it would be spaceflight and ground control. It's just starring Kennedy Space Center because for the data that we're going to work with in the hands-on portion, it's going to come from the Rodent Research 1 mission in which mice were either flown on the International Space Station or kept in a chamber on Earth at Kennedy Space Center. And then you're going to take your sample from your organisms here. In our case, it was the soleus muscle, which is one of the muscles that make up the quadriceps. You're going to isolate your RNAs from all of your samples. And after you isolate your RNA, keep in mind from our sequencing overview lecture in the GL4U intro module set, it's important for RNA sequencing data that you remove ribosomal RNA. And so we talked about different ways you can do this. One is to target and remove your ribosomal RNA, either with an enzymatic approach or a hybridization pull-down approach that we talked about previously. Or you can positively select for the poly A tail that exists on mature message RNA to be able to enrich your sample for message RNA. After you remove your ribosomal RNA by either mechanism, you're then going to move on to build and construct your sequencing libraries. This consists of adding on your adapters. As a reminder, 3 critical pieces to your adapters. The first flanking your target region is going to be your sequencing primer. The second is going to be your index. Your index, again, is going to let you know which sample each sequence came from. And then that third region is going to be either your P5 or your P7, and that's going to allow you to hybridize to the flow cell to be able to do the sequencing. After you have your library successfully constructed, very critically, you're going to perform your library quality control assessment. If you recall from the sequencing lecture of the GL4U module set, we are checking for 2 things during library QC. We are checking to make sure we do not have primer dimers. As a reminder, primer dimers are when 2 adapters or dimers, or 2 adapters come together to form a dimer without having a target sequence in the middle. These are very dangerous because the flow cell has a bias towards short sequences. And so these primer dimers are going to be more likely to latch onto your flow cell and get sequenced over your longer libraries that have your target region of interest. And so even a small amount of primer dimer contamination, as little as 5%, can end up taking up about 60% of your reads. And so very critical in this library QC step to make sure your libraries don't have any primer dimers or adapter dimers. Also, you want to check the size range of your libraries to make sure it's consistent with the length that you're going to set your sequencer to do the sequencing, keeping in mind that your insert length should be such that if you're doing paired end sequencing, your forward and your reverse read will have a little bit of overlap. So if you're sequencing at paired end 100, which means 100 in your forward direction, 100 in your first direction, your insert or your target that you're going to sequence of your cDNA, you want to aim for around 170 base pair targets. You get about 30 base pair overlap. And then keep in mind when assessing the size of your libraries, you need to account for the size of your adapters that are on either end. And those will be roughly 65, 70 base pairs, depending on how long your index is. And so if you have 65 base pair primer on either end, that's 130 base pair total, plus the size of your insert at roughly 170 base pairs. For paired end 300 sequencing, you are paired end 100 sequencing, excuse me, you are looking for a library size range around 300 base pairs. Again, your insert plus your adapters. Once you confirm that your library sizes are what you want, you don't have adapter dimers, then you're going to quantitate your libraries to know how much to put on your flow cell, and then you're able to load your flow cell and perform your sequencing steps. The first thing it's going to do is undergo cluster formation. That's where it's making millions of copies of your libraries in each spot on your flow cell. And then it's going to go through and do sequencing by synthesis that we covered in the GL4U introduction module set. Once you're done with your sequences, the next thing that you are going to do is assess those sequences for low quality sequence data and determine if there's any adapters in there. We did talk about this in the GL4U module set and that hands-on sequencing QC Jupyter notebook at the very end of the GL4U module set. We will review that in this module set. But that's what you want to do is check the quality of your raw sequence data, remove any low quality sequencing data, remove any adapters from that, and then you can go on to map those trimmed filtered reads onto your reference genome to start to determine or figure out where those genes came from, which genes are expressed in your samples. And you can then take that and perform downstream analysis, like we'll do looking at differential gene expression and some visualizations. So a little bit about the RNA sequencing parameters. So sequencing length, again, this is determined by how many cycles you set your sequencer to. Longer reads is going to increase your gene ID confidence because your genome has a lot of repeats in it. The genome of many organisms have a lot of repeats in it. And so the longer your sequences are when you're mapping it back to the reference genome, the longer it is, the more likely it is it'll only map to one location, which will increase your confidence that that's the gene that was expressed, that that's where that RNA came from. However, if you have shorter reads that you're mapping back, you increase the likelihood that it'll map to multiple locations on your genome, and then it'll make it more difficult to determine where that RNA came from originally or which gene it is. Our standard at GeneLab for bulk RNA-seq is 150 base pairs. We do 150 base pairs paired end. Sequencing depth is another thing you want to consider. Sequencing depth is the number of sequences per sample that you generate. So the greater the depth, it'll increase the likelihood of sequencing low abundant transcripts, detecting novel transcripts, and quantifying isoforms. So as a reminder, in the GL4U intro module set, I used the example of having a container filled with mostly blue and red M&Ms and just like 5% green M&Ms. And so if you want to get one of those green M&Ms and you're just pulling out M&Ms randomly, you're going to have to pull out more M&Ms to increase your likelihood of reaching a green one, which would represent a low abundance gene in this analogy. Also, keep in mind that you're going to want to sequence at greater depth if you prepared your samples using the ribodepletion method over the poly-A method. And this is because if you did the ribodepletion method, if you recall from the GL4U intro module set, that means that you have more types of RNA left in your sample. So if you wanted to do something like differential gene expression, looking at your messenger RNA, you're going to have to sequence deeper to make sure that you get a sufficient sampling of the message RNA that's expressed in your different samples. Whereas poly-A enrichment, you are enriching your sample for message RNA, so that should make up the majority of those sequences in your sample, and so you don't have to sequence as deep. In the GL standard for mammals, prepared for ribodepletion is around 60 million reads per sample. Prepared with poly-A selection is around 40 reads per sample. And also, as a reminder, more biological replicates are usually preferred over greater depth. You really want to increase your N to be confident in your downstream assessment when you're comparing two groups. And paired end is often preferred over single end. And this is because, as we talked about in the GL4U intro module set, the efficiency of shorter reads, or the quality of shorter reads, is going to be better than longer reads because as you increase the amount of sequencing, the efficiency of your enzyme will reduce, and the quality will reduce the longer your read is. So paired N150 sequencing is going to be preferred over single end 300 base pairs of sequencing, because you're just going to see better quality in your reads. So now that we've looked at or discussed some RNA sequencing parameters and did a little overview of an RNA sequencing experiment, let's talk about how we process those data. So the first thing that we're going to do, as we did or looked at the end of our GL4U introduction module set, is assess the quality of our sequencing data. So looking at those raw reads and checking the quality using that FastQC program that we discussed previously. And so raw data generated from the sequencer, as a reminder, are stored in what's called FASTQ files or base call files, which are then converted to FASTQ files. So for the purpose of this module set, we're going to be starting with FASTQ files. And just a reminder, once you're at FASTQ file, that conversion of base calls into FASTQ files will utilize that index read to be able to separate your samples, all the sequences from one sample, into separate FASTQ files. So you should have separate FASTQ files containing all the reads for each one of your samples. As a reminder, FASTQ files contain multiple reads, and each read is formatted with 4 distinct lines. As a reminder, again, we covered this in the intro module set, but just as a reminder here, the first line is going to begin with an @ symbol, followed by information about the sequencing run, such as the sequencing platform, the run number, flow cell ID, and cluster location, also the read number and/or the sample index. So it is true that some databases will change the information in this first line of your read. It is always going to start with an @ symbol, but it's good to have this information here so that way you can identify the address that on the flow cell that this sequence came from. And then here we see a 1 in this position here. That indicates that this is a forward read. If it was a reverse read, we'd probably see a 2 there. And then you see this 6 base pair sequence here is indicative of the index that is used to specify this sample. So that means that every single read in the FASTQ file for this sample, we're going to see that same exact index sequence because that's what was used to separate out these reads for this sample. Line 2 is going to contain the actual target sequence here. So this is going to be written as A, T, C's, and G's. And just as a reminder, the sequence length is going to be equal to the number of cycles that you programmed your sequencer to run. That third line is just a separator line. It begins with a plus sign. There could be other things written here. I don't think I've ever seen a FASTQ file where other things were written in that third line, so it's primarily used for a spacer. Always starts with a plus sign. And then that fourth line are going to be quality scores of each base call that are for red 33 encoded, and they use ASCII 2 characters to represent the quality of the bases. So you'll see that there is a encoded letter for every single base in our sequence to represent the quality score, which we're going to talk about next. So this is looking here at the ASCII 2 base 33 mapping for the quality of each nucleotide call in the sequence. And so as a reminder from the GL4U intro module set, the higher the quality score or the Q score, the more confidence you have that if the sequencer called a base an A, that it actually was an A. And so you could just see here from a quick look that these higher Q scores tend to be encoded by capital letters in the ASCII 2 mapping, whereas lower quality scores are represented by symbols or even numbers here in the ASCII2 mapping. And just what that confidence looks like here, as a reminder, this is in a logarithmic scale, so that a Q score of 10 is going to represent a 1 in 10 probability of the base call being incorrect, or a 90% accuracy. A Q score of 20 is representative of 1 in 100 probability of an incorrect base call, or a 99% call accuracy. A Q30 is highlighted here, which is encoded by a question mark, is going to be representative of a 1 in 1,000 probability of an incorrect base call, or a 99.9% accuracy on down. And so, of course, reading all of those quality scores and having to map that back from the ASCII2 encoding to the Q score, and then looking at it for a length of 100 plus bases for these FASTQ files can contain millions of reads. It's pretty impossible to be able to assess quality looking at these things one at a time, which is why this FASTQC program is so great. It's one of the most common ones used to assess the quality of sequencing data. I know we went through this in the GL4U intro module set, but we're going to review it again here. And so this FASTQC program will give you a report after it's done, and each report has specific modules looking at different aspects of the quality. So one module here is going to be the basic statistics, which will tell you the name of the file that it's evaluating, what it sees in the file type, what encoding was used for the quality scores, the total number of sequences or read depth, the sequences that were flagged as poor quality based on the metrics within FASTQC, which, as we talked about before, needs to be taken with a grain of salt, because you need to understand what sequencing data you're assessing to know if it's good or bad quality, or what to expect. Also, the sequencing length here, again, for raw data, this should be the number that you set the number of cycles to when you programmed your sequencer, and then the average percent GC across all reads in your particular FASTQ file that you're evaluating. So in this example here, are these reads forward or reverse? And you can see from the file name that there's an R2 in the name, so that would indicate that this is a reverse set of reads. So we assume that this is paired-end reads, and this is the reverse set of reads. Because there's this R2 in the file name, it's pretty common to put either an R2 or a 2 in the file name to represent reverse reads. However, people can change file names, so that's not always the case. For the data hosted on OSDR, we do make sure that for paired-end reads, we always have an R2 to represent reverse reads and R1 to represent forward reads in our file names. What is the read depth for this sample? So again, the read depth is the total sequences of your sample. So here we can see that it's 100,600,211 reads are inside this FASTQ file. What about the sample read length? And here again, the sequence length you'll see is 150 base pairs. You'll also notice that this is one number. That means that every single read in this FASTQ file, all 100 plus million of them, have the same length of 150 base pairs. This is actually a good way to know if this is raw data, because this should all be the same, or it could be two values, one all the same value. Oh, sorry, no, this will always be the same for raw data. If there's been any kind of manipulation or trimming going on, this will be a range, as we'll see later. Oh, and I do want to point out that the link to the FASTQC program for more information about these module sets is going to be at the bottom of all of these slides. So the next module, FASTQC module we're looking at here, is the per base sequence quality. So this is going to show the position of the read on the x-axis here, and then the Q-score on the y-axis. Really nice because it did the conversion of those ASCII 2 characters into Q-scores. And as we remember for both the intro module set and earlier in this lecture, the higher the Q-score, the higher the quality. And so question here is, do you notice anything about the quality as you move towards the end of the read? And so you can tell here that the quality does start to drop a little bit at the end of the read. You also want to mention these are box and whisker plots where this red line is representing the median value. This blue line is representing the average value at each read position across all reads. And so, yeah, we're seeing this quality decrease. That's pretty common. Also, these data are for the reverse reads. Do you think that the quality would be different for the forward reads? So although you may not think so, the quality of the forward reads is often better than the reverse reads. We're not entirely sure why, but it may be due to the fact that the reverse reads are sequenced after the forward reads, and therefore the template cDNA and or the other reagents may have already degraded slightly, reducing the quality of those reverse reads. Next thing we're looking at here is the per sequence quality scores. So this is the average quality score or Q score, and these are the bases throughout each read. And so looking at the quality of all these bases, how you interpret this is if you want to know how many base calls had a Q score of 33, you would just 33, go up here and then move over to the y-axis and you'd see that value of the number of bases with that quality score. And so would you consider these data to have good quality overall? Yeah, for sure. So it's definitely the majority of the base calls made of the reads in this FASTQ file have very high quality. So yeah, I'd say yes. Next here is the per base sequence content. And these values should be fairly consistent across the read length in an unbiased library. So unless you specifically over-enriched a certain gene or something, or did some kind of manipulation to your samples, usually for bulk RNA-seq, we will see that this is a roughly unbiased library, except for at the beginning here of the read. Recall why this could be is, remember, when we are converting our RNA into cDNA or complementary DNA, we use a primer to initiate the reverse transcription, and that primer is often a random hexamer, and that just means 6 random base pairs. And so the idea is that those 6 random base pairs will unbiasedly start reverse transcribing that RNA and to ensure that all the RNA is reverse transcribed. However, although that hexamer should be random, there's always going to be some bias in biology. And because of that slight bias, we do see that slight bias show up in this per base sequence content for those first roughly 6 or so base pairs. And if the library is biased, you're going to see spikes in the data. Again, we see some here. They could be even worse, as we saw in the Jupyter notebook for the amplicon data in that introduction module. The nature of RNA-seq primers, as I mentioned, tend to cause some wobble at the beginning of the reads, like we see there. The next module I'm going to go over is the per sequence GC content. And for this, we expect the GC content to be normally distributed, which is going to be represented by this blue line here. Our actual GC content data is in red. And so the question here is the GC content normally distributed across all reads, represented by this red line? For this data set, kind of, right? It kind of follows this theoretical distribution. However, we have a couple of peaks here, and we may just have some regions of the read that's going to have a higher GC content, like we're seeing with these peaks. However, we do see this small peak over here that has a higher percent GC. So what do you think that the peak outside of the normal distribution could indicate? It might indicate some level of contamination, whether it be ribosomal RNA, some primer contamination, or maybe even some other type of contamination. Next module here is the per base end content module. And so if the sequencer cannot confidently determine what base is being read in the sequencing by synthesis process, it will go ahead and just label that base as an N, which again means that it can't confidently say that it's one of the 4 bases. And so you'll see an N in your sequence data. And as you can see plotted here, this is going based on the position of the read for all reads in that FASTQ file that's being evaluated. And this is the percent N content in each one of those positions. So how often was a base able to be called in these reads? So not very often at all. You see it's pretty much 0 across all positions, just very rarely, maybe 1 or 2% of the reads at that position 1 was an N. Next, we're looking at the sequence length distribution module of FastQC, and this is showing the sequence length on the x-axis and the number of reads here on the y-axis, and we see that the length of all reads are the same, 150 base pairs, as they should be if you're looking at raw data. So are all reads the same length? Yes, they are. What is the read length for these data? I already mentioned that, 150 base pairs. Next, we're looking at adapter content. This is to determine if there are any adapters in your sequence data. As a reminder, if your sequence of interest is shorter than the number of sequencing cycles, you will sequence into your adapter. So just a quick look at what that looks like here. So in this example, we have our insert region represented in green here of 130 base pairs. Flanking that, we start getting into our adapter, right? Our sequencing primer, our index region, and our P5 and P7. And so as read 1 sequences through here, it's going to start right after that sequencing primer, right? That's going to recruit polymerase, and polymerase is going to start sequencing here. However, we program the sequencer to sequence at paired end 150 base pairs. So it's going to go ahead and sequence through your 130 base pair insert, but it's still going to continue on the full 150 cycles, which means that it'll start sequencing into the adapter on the other side. And that's how we end up getting adapter sequences in our reads. Similarly, if this is paired end sequencing, the reverse read, again, will start at the sequence priming region of the opposite end here, begin to sequence through your target region, right? And then again, if the number of cycles is more than your insert, as it is here, it will go past your insert and start to sequence into the adapter region on the opposite end. So you notice here for both the forward and the reverse reads, we start by sequencing our target region. And again, if your sequencing cycles are longer than your target region, it'll go into the adapter sequence at the end of that read. Important to keep that in mind as we're evaluating this. So does this example contain adapter sequences? Yes, it does. We can see that it contains adapter sequence, and by default, FastQC has a few different common adapter sequences that it scans for. And so this here, it is detecting the Illumina universal adapter in your sequences. And where are the adapters relative to the read position? They're actually at the end of the read. Again, because the sequencing is going to start from that priming region, so it will always start, unless you're sequencing an adapter dimer, it will almost always start at the start of your template. And so any adapters are going to be found at the end of your read. Do you think that this will be a problem? Probably, right? Because we added on these adapters artificially, and ultimately we're going to be mapping these reads back to our reference genome. So if the reads didn't come from biology, they're not going to be able to map to our reference genome. So we're probably going to want to remove these before we do any alignment. The next module here is the sequence duplication module. As a reminder, these data are based just on 100,000 read sampling of the total number of reads, and it only evaluates the first 50 base pairs. So if you have 150 base pair reads that you're evaluating here, this module is only going to look at the first 50 of those, and it's only going to look at the sequence of the first 50 bases of the first 100,000 reads, and then compare those to all the other reads to see if there's any duplicates. And so it's usually an overrepresentation of the number of duplicates. Also, if you remember from the intro sequencing QC Jupyter notebook, these duplicates detection here is not going to be able to distinguish between biological and technical duplicates. And so this module is going to be used to determine how unique the reads are. The blue line indicates the percent of duplicated reads at a specific duplication level, right? So this is indicating the number or the percent of total sequences that have a duplication level of greater than 10 or more than 10 copies. Oh, and the red line shows what these data would be if you were to deduplicate or remove duplicates from your data. As a reminder, reduplication can be either biological or technical in origin. Biological is, of course, if you have a gene, for instance, that's highly expressed, there will be multiple copies of the transcript for that gene in your sample. And so when you are chopping up your RNA during library preparation, it is possible that you can cut multiple copies of that same gene in the same location, which will end up giving you the same sequence. And so that will have come from biology, and those types of duplicates we would want to keep. Technical duplicates, as a reminder, can come from the nature of the flow cell. It can also be the result of that final PCR step in your library preparation. And we'll talk a little bit more about duplicates in the technical duplicates in the next slide. And so if you have a very diverse library where all reads are unique, you would have 100% of reads would have a duplication level of 1. Of course, I would be very wary of that because I would want to make sure that might mean that you didn't sample everything in your sample. You didn't sample all the sequences in there. And so I always do like to see some amount of duplication. The amount of duplication will depend, of course, on the sequencing assay that we're performing. So it's common specifically when working with enriched libraries to have some level of duplication. Again, if it's enriched, that means that you are looking at a particular region of the sequence, so you would expect it to have duplications. Lower duplication levels in the 2 to 9 copies could be likely derived from, well, either PCR technical duplications, or it could be from interesting genomic regions. So again, this is if you have multiple copies of one transcript in your sample, your original sample, and then when you do your chopping, it chops multiple copies in the same place. You'll have the same sequence again from biology. Moderate duplication levels, 10 to 100 copies, could be reads that were derived from ribosomal RNA or some kind of highly repetitive genomic region. And really high duplication level, more than 100 copies, could be a library issue, either adapter dimers, or it could be too much Phi-X. I didn't mention this previously, but Phi-X is used as a control that's spiked into libraries just to ensure that there's enough diversity in your libraries to be able to distinguish signals from different base pairs. So are duplicates present in this sample? And should we be concerned? Yes, and yeah, probably if they're technical duplicates, we want to get rid of them. And roughly what percent of reads are duplicates. You can know this by looking at the percent of sequences remaining if it were deduplicated. So basically 100% minus this number will give you the percent of duplicates at the start, which is around 73.5% in this sample. Keep in mind taking it with a grain of salt because this is just a sampling of all the reads in your FASTQ file. Just to quickly go over some ways to get technical duplicates. One of them here, there are two different types of flow cells for Illumina sequencing. There is unpatterned and patterned flow cells. Unpatterned flow cells are those that you saw in that Illumina sequencing video at the end of the last lecture of the GL4U intro module set, where the clusters are not separated by a barrier on the flow cell. There's just a lawn of those P5, P7 complement regions. And so what can happen is during cluster formation, one of your pieces of library, so one of your strands that you want to sequence could latch on, hybridize to a P5, P7 in the cluster area, but then fold over into a nearby cluster region and also populate that one, in which case you would have two adjacent clusters that were derived from the same original sequence. And so your scanner here could interpret this as two separate clusters, even though it came from the same original sequence scan. So that would be an optical duplicate. Something similar can happen in pattern flow cells where one strand is populating one location within your pattern flow cell, but then folds over into the adjacent location and forms a cluster in that one as well. And so again, deriving from the same sequence. So this would be a technical duplicate. Also down here, these types of duplicates happen on both unpatterned and patterned flow cells. One, as we talked about, could be a PCR duplicate. So at that final step in library preparation, we amplify the amount or make copies of our libraries using PCR. So the copies that we make are technical in nature. We have made them doing PCR. And so we can sequence the copies that we made artificially in that PCR step, in which case it would show up as the same sequence in two different locations, but that came from a technical artifact, that PCR amplification. The other thing that could happen here is if complementary strands from your sample library populate two different locations on your flow cell, then again, you have the same origin sequence populating two different clusters. And so that is another form of a technical duplicate. These duplicates here can be controlled for by adding those universal molecular identifiers or UMIs prior to the PCR step that I talked about in the GL4U lecture, a GL4 introduction lecture. And then if you add those UMIs, which are going to be unique for each sequence prior to the PCR step, after it undergoes PCR, then if your target sequence is identical with another target sequence, but they have different UMIs, that means that that duplicate came from biology. But if your target sequence has the same sequence as another sequence, and both of those sequences also have identical UMIs, that means that that is a technical duplicate and you can go ahead and remove it. Also being able to remove these optical or clustering duplicates. This can happen so long as you have the address of that cluster in the read header. Then you could see if you have the same sequence that is in adjacent regions on your flow cell, you can identify those and remove those technical duplicates. Do you want to say for the GeneLab standard pipeline, we do not currently remove technical duplicates. It's actually not a common practice in bulk RNA sequencing. However, we are looking into being able to remove duplicates with data sets that have UMIs in them. And so that might be a future addition to the GeneLab RNA-seq pipeline. But for now, we do not remove technical duplicates. So once you've assessed the quality of your raw data, the next thing that you're going to want to do is trim and filter your raw data. So you're going to want to remove any low quality sequences from your data. You're also going to want to remove any adapters that you identify in your data. And lastly, remember, if you have really short reads, you increase the likelihood that it's going to map to multiple locations on your reference genome, so you won't know where it came from. And so another part of the filtering step is going to be able to get rid of anything that's just going to be too short to be able to confidently identify where it came from. So for us at GeneLab, we use a program called Trim Galore to do this. So let's get into a little bit more about trimming and filtering data. So why would we want to trim or filter? This is also known as pre-processing raw sequencing data. Well, as we talked about, we'd want to remove any low quality reads. We'd want to trim off any adapters because we added them artificially, so it's going to affect our mapping efficiency. We didn't talk about this yet, but we might want to trim 3' or 5' ends as well. Just as an example, keep in mind that we went over this at the end of the GL4U introduction lecture, but when we are creating mature message RNA, remember the first step coming from DNA, it is transcribed into pre-mRNA. And then after it's pre-mRNA, it undergoes some RNA processing such that, if you recall, there's some splicing events that occur. And then also we see a 5' cap added on at the 5' end and a poly A tail added on at the 3' end. And so that poly A tail and 5' cap, it did not come from our DNA template, right? So when we go back to map to a reference genome, we wouldn't be seeing those pieces in our reference. And so another thing that you can do is, if you're detecting poly A's in your sequencing data, you can go ahead and chop those off in your trimming. And also, lastly, just talked about this, we're going to want to remove any reads that become too short after trimming. So let's say we remove poor quality, we remove adapters, and it just so happened that our adapters and our poor quality data took up a big chunk of our read length. And so we end up being left with like 15 or so base pairs after that. We're not going to want to keep that around because it's just going to map to too many locations on a reference genome. We're not going to be able to figure out where it came from. So we'll just want to filter and get rid of that. There are several tools to do these pre-processing and filtering steps. One of them is Cutadapt. There's also Trim Galore, which is the one we use on the GeneLab pipeline, and Trim Galore actually uses Cutadapt under the hood. Another tool is called Trim-O-Matic. There's also HT-Stream you could use to trim. This is not comprehensive by any means. There's a ton of different trimming tools. Most of them work roughly the same. And so these are just some parameters that we use for the GeneLab pipeline with Trim Galore. We let the instrument know that our quality scores are FRED 33 encoded. We also want to tell it what Q score to use to trim reads that do not meet that threshold. We actually use the default, which is a Q20. So anything that is going to be less than a Q20 going to get chopped off of our reads. We also can tell it what length we want to keep. So again, if we're trimming, we want to tell it what the minimum length is to keep. We use the default of Trim Galore at GeneLab, which is 20 base pairs. So anything less than 20 base pairs is going to, any reads less than 20 base pairs is going to get removed. We also want to tell it that we have paired end data. If we have paired end data, if it's single end data, this option is removed. Why this is important is because if you have paired end data, when you do any downstream tools like alignment or your quantitation, and you tell it that you have paired end data, it is going to expect that for every single read in your forward FASTQ file, so your forward reads, it is going to have a mate pair, a reverse read in your R2 FASTQ file, right? In your reverse FASTQ file. And so when we're trimming, you saw that the quality of your forward and your reverse reads can be slightly different, right? And so as we're quality trimming and removing adapters, let's say that our forward read, once we do all of our trimming, we get that down and we end up being left with 22 bases, right? And then that respective reverse read, we quality trim that, remove adapters. But after that's all said and done, we're down to 18 base pairs. And we have a threshold length of only 20 base pairs. And so if we didn't set this paired end flag here, it would just remove the reverse read, but it would keep the forward read. And so then when we put this into subsequent tools for analysis and we tell it that it's paired end, it's going to give us an error because it's going to say, no, we found reads where there's a forward read, but no reverse read, and it won't be able to work. And so by adding this paired flag, what we're telling Trim Galore to do is that after you trim all the forward reads and all the reverse reads, if you have the case where either the forward read or the reverse read does not meet that 20 base pair length cutoff value, then you just get rid of both the forward and the reverse read for that read mate pair. Also, you'll notice we don't specify our adapters here. If you don't specify the sequence of your adapters, there are some common adapters that are pre-programmed with the Trim Galore and Cut Adapt tools. And so it will automatically scan your reads and detect which adapters are there. And then it'll use that information to remove those, to identify, remove those adapters in your reads. After you do all of your trimming from your raw data, you're going to be left with trimmed reads, trimmed and filtered reads, right? So the next thing you want to do is perform the FastQC analysis again, this time to check the quality of your trimmed reads. And so this, we're just going to compare some of the modules, not all of them, between our raw data and now our trim data. So just as a reminder, pulling up the raw data for this FastQ file we were looking at earlier, comparing it with the trimmed data FastQC report here. So how many reads were removed during the pre-processing or trimming step here? Well, you could just look at the total sequences of your raw data and then subtract the total sequences left after trimming from that number. And you see here that we have around 146,000 reads that we ended up removing after trimming. What's the sample read length after trimming? So here we're going to look at the sequence length parameter, and after trimming, we see that this is a range from 20 to 150 base pairs. So this is a range meaning that the reads in that FASTQ file can range from as low as 20. Why is it to go down to as low as 20 and no lower than that? Because we set a cutoff of 20 base pair read length. So if it was less than 20 base pairs, after trimming, it would have gotten removed. So that's why the lowest we see now is 20, and the maximum read length is not going to exceed the number of reads in our raw data, which was 150. So that's why we see that range here. Just taking a look at the per base sequence quality scores, our raw data on the left here, trimmed filter data on the right. And here, how was the per base sequence quality changed after trimming? Well, we can see that the quality improved here. Next question is, what would happen to the quality if a Phred score cutoff of more than 20 is applied? Well, if it was quite a bit more than 20, right? So let's say it was a Q score of 28 was our threshold that we would use, we would end up seeing that the post-trim reads would have even better quality than this here. Last module we're going to look at is the adapter content. Again, it's important to make sure that you check this. And so after trimmed or filtered data, what happened to that adapter content? The adapters went away. And you really want to check this, especially if you specify a particular adapter in your trimming step, and then you do this QC assessment and you're seeing that another adapter is still there, you're going to want to retrim to make sure you get rid of those adapters. And so that brings us actually to the end of part 1. And so before we get into the alignment step, which is our next step after our pre-processing or trimming, I'm going to pause here and have you guys go and start the RNA-seq processing Jupyter notebook of the hands-on portion of the GL4U RNAseq content. And you're going to go ahead and run the steps in that notebook through the end of your trim data QC, and then come back and pick up with part 2 of this RNA-seq overview lecture. I'll see you guys all then.